Hello, good afternoon, everyone. We are going to start on lecture nine today. And as I mentioned before, lecture nine is pretty complicated. There are a lot of complex concepts that we have to go through. So please feel free to stop me if you need me to clarify certain things along the way. Okay, so lecture nine is called biotechnology. What is biotechnology? Biotechnology, as the name implies, is essentially technology that is based on biology. So, up to this point of the course, we've studied various biological processes. We understand how cells replicate themselves. We understand how DNA are being copied. We understand the process of protein production, like from the DNA to RNA and then to proteins. So after scientists understand these biological processes, they are trying to control these processes. They try to alter it, change it in a way that would harness the power of these cellular and biomolecular processes so that you can use it in a way that you want it to work, and often at the end, these kind of manipulations is going to create technologies and products that will help improve our lives. So I'm going to go through a few examples of biotechnology with you. There are a lot of different types of biotechnologies out there. It would be like a course on its own to go through each one of them in detail. So, we're not going to have enough time. I've selected a few common ones to go through with you, a few easier ones to wrap your head around, and from that, you can see how it relates to some of the stuff that we talked about throughout the course. Now, like I said, we are nearing the end of the course. This is lecture nine. There's only one more lecture. This is week 12. And then week 14 is the final week, which is when you were going to have your final test. Okay. So just hang in there. We're going to start with this concept of recombinant DNA. DNA. And it's a type of biotechnology. Recombinant DNA basically refers to fragments of DNA that are as a result of combining DNA from multiple organisms, from multiple species. Yeah, so you can take some DNA from jellyfish, and you can combine it with DNA from the mouse. And what happens is you end up creating these glow-in-the-dark mice. They are called GFP mice. You can Google it if you're interested. It's for real. They do exist. Okay? It's a combination of jellyfish gene with mouse genes. Okay, so now you have a recombinant DNA because the end result consists of genetic material from different species. Okay. Now, why do people make these glow in the dark mice? Like, what do we use them for, right? It turns out they are actually very important in research. A lot of medical research uses these GFP mice as a model in some cases. But we're not going to go into the details of it. You can look it up if you're interested. So I will give you two examples of recombinant DNA applications, one in medicine, the other in agriculture. Okay. Once again, just in case you missed what I said, recombinant DNA is basically just DNA that you create using genetic material from multiple sources. Okay, you can take it from a flower, and then you can put it, you can put bacteria DNA together, and then now you have recombinant DNA. It doesn't even have to be two sources. You can have three sources, four sources, so on and so forth. Of course, the more sources of DNA you incorporate, the more complicated the technique is going to be. But I think we are at a point where generating recombinant DNA is it's pretty straightforward. Okay, the technology has matured. So I'm going to start with the agricultural application for recombinant DNA first. That's the easier one to understand. The medicine one probably going to take us the whole class, to be honest. So let's get on with it. Has anybody heard of golden rice? Just wondering. Right? Just give me a thumbs up or, type in the chat box if you have heard of golden rice. Anyone at all? Okay, well, if you haven't heard of it, this is what it is. Okay? First of all, in poorer countries, low-income countries, you can say, right? Vitamin A deficiency is very common. Okay? Vitamin A deficiency. Now, we don't think about it too much in North America because, it's rarely a concern for us. But if the body does not absorb enough vitamin A, it could have several severe outcomes, right? Including causing blindness in kids. Okay, these are preventable childhood blindness because the problem is caused by vitamin A deficiency. So if you introduce vitamin A in their diets, then it's preventable. Right? Also, vitamin A deficiency can lead to lower immunity. Right? And in these developing countries, low-income countries, there is often low vaccination rate as well. So if the vaccination rate is low and the community has low immunity, these two combinations it doesn't go well together. It could spark endemic for infectious disease. Now, you all know what a pandemic is. Pandemic is global, right? Okay. And then you have an epidemic, which is a regional thing. Okay. And if you go a little bit slower, like within a community, and you have these outbreaks, then we call them endemic. Right. So endemic is the lower scale. It's often within a community and adjacent areas. It's relatively contained, but it could have a potential to spiral out of control if it does, spread further than that. Okay, so vitamin A deficiency can cause quite a bit of problems. In some countries, each year it is estimated vitamin A deficiency is responsible for 670,000 death in children under the age of 5, and it causes an additional 500,000 cases of irreversible childhood blindness. Okay, just quick reason, the vitamin A is needed to make a particular pigment that allows you to see in the eye. If you don't have that pigment, you can't see. So that's how it causes blindness. Here's a heat map that shows you where the vitamin A deficiency is the most severe. And you can see it's not too much of a problem it's not a problem at all in Canada or in the United States. But once you get into some of the lower income countries, you can see it is a quite severe a problem. Okay, so what's the problem? Well, the problem is there are no access or limited access to vitamin A rich diet in these countries where the problem occurs. It's difficult to obtain these food, and it's not always feasible to transport um, high vitamin A food to those places, okay? Because they might get spoiled easily during transport, and sometimes it's just, outright expensive. Um, what do people tell kids to eat because it's good for their eyes? Like, have you heard that before? Like, I hear
Carrot?
that's why they say carrots, right? Because carrot actually contains something called beta carotene. That's why carrots are orange, okay? And beta carotene is a building block for vitamin A, okay? So, if you eat more carrot, you would get your vitamin A. There are other food that are rich in vitamin A, rich in beta carotene as well, which allows you to build vitamin A in the body. But again, it's hard to get some of these food and mass distribute them to these countries. Okay, so that's why they create. Can you give some examples of those food? Basically anything that is a vibrant orange or yellow in color, like peppers, bell peppers, for example, right? They are pretty good, high in beta carotene as well.
Pumpkin can be?
Yep, pumpkin, absolutely, right? Yeah, so anything that's orangey, yellowish, red, they are pretty good in beta carotene. So my point is, you cannot always get these food to those countries that need them. And so that's why we need to have a solution for this problem, which is where golden rice comes in. Okay? Golden rice is genetically engineered rice. Regular rice, as you know, is whitish in color. I mean, some rice are darker in color than others. Brown rice, for example, is a little bit darker. But rice, regardless of what kind you consume, does not naturally produce beta carotene. Okay? Golden rice is genetically engineered rice that contains recombinant DNA. Okay? And it allows the rice to produce beta carotene. To make golden rice, they took a gene from daffodils, which are these flowers right here. The gene is called Psy. When they write the names of a gene, they use italics to show you. So there's a Psy gene from daffodils, and then there is another gene called CRTL from a kind of bacteria that lives in the soil. And they put these two genes into the genome of the rice, and that allows the rice to produce beta carotene. Okay, so now we have got the golden rice rich in beta carotene, right? So as you can see, in a lot of these places where vitamin A deficiency is a problem, rice is actually a staple food for them, right? They have a lot of rice in their diet, anyways, and rice is relatively easy to transport without worrying about getting spoiled. Okay, rice can stay fresh for reasonable amount of time as long as you keep them dry, right? So it's much better, easier to transport, and more economical to transport compared to something like carrot. And we talk about like carrots and pumpkins, right? Which again, even if you get them to these places, people might not uh, enjoy eating them, right, as part of the diet. But if it's something like rice, they already eat them anyways. So, switching it to like a different version of a rice, I mean, from what I read, they don't taste like carrot or anything, okay? It's taste like regular rice, but they are rich in the beta carotene, okay? And that would solve the problem, right? Now that they eat the rice, they get their beta carotene, they can make vitamin A in the body, and that should help prevent no low immunity as well as blindness. Are you guys with me so far? You follow the story? Yeah.
Yes, it was
Uh-huh.
interesting, by the way.
Mm. Thank you. Now, so is this a good thing or is this a bad thing? Well, as with many things, there are two sides of the story, and here are some, basic controversies that you can look up if you want to read more about it. People who support the idea of golden rice, I'm sure you can come up with the arguments. First, it addresses the vitamin A deficiency problem, right? If you can prevent a kid from going blind, why wouldn't you, right? That's what they're saying, right? It would improve public health because don't forget, vitamin A deficiency lowers your immunity and you can be more susceptible to getting sick from infectious disease. So, that's gonna consuming these golden rice is gonna improve not just individual health, but collectively improve public health and therefore reduce healthcare costs, right? And also, it can enhance food security. If you can get a lot of these rice stock up in these places, then it's gonna potentially fix a hunger problem as well. But not everybody agrees with this approach. There are opponents of golden rice saying, for example, there could be unintended consequences. Okay. We are playing with nature here. Okay. We are playing God, some say. You are putting genes from different sources together into an organism. Um, sure, short term, it doesn't seem like there are any effects, but who knows what's going to happen long terms, right? Like, is it safe to consume these things over long periods of time? Like, what if these genetically modified plants gets out into the wild? Is it gonna affect the ecosystem? Is it gonna take over native species that are growing on their own? Like, it's gonna create competition, for example. Like we don't know. They're saying, right? Um, it's kind of like this, right? When people first started smoking, they didn't know it was bad, and then 10, 20 years later, finally they realize, oh, it has a negative effect on it, right? You know what? I read something interesting recently. Just this is like on a side note, totally kind of related to this, but not really. Um, and this is true. You can look it up. Okay. you guys all know X-rays are bad, right? Okay, like sure, you get your X-ray at the dentist, and then you get your chest X-ray sometimes if you have a lung infection. So small doses of X-ray is okay. It's not gonna be like super dangerous for you. Uh, but if you constantly get exposed to X-ray, like it's going to increase your risk of getting cancer substantially, right? Okay, so X-rays are bad. But I recently read that back in the days in shoe stores, okay, stores that sell shoes, shoe stores, I guess, they have these X-ray machines. And the kid or whoever's buying the shoe, they would stick their feet in, and then there's like a little peek hole that they can look through to see the X-ray of their feet, with the bones and everything. And the idea is to see, how your feet fit within the shoes to see if it has a good fit or not, right? And I read that I was just this is crazy, right? That like so many people are gonna get like overexposure to X-ray, and we're talking about kids there, right? So that's what I mean here, right? Unintended consequences. Just because something sounds like a good idea at the moment, it might not be a good idea after we have more understanding of the situation. And that's what the opponents are saying. Okay. So far there are no unintended consequences that has been documented for golden rice. Okay, but I can see why this is a valid point. All right. Anyways, another argument is that there are corporate control and patents. Obviously, these are very advanced technologies, and companies spend a lot of money, coming up with these engineered products, and a lot of them have patents on it. And so people are afraid that, these corporations have too much control of this product, which is now they're supplying to developing countries. They worry all these a corporation has too many influence on those countries, okay, including perhaps how the government runs, because now they have too much power. They say, okay, unless you do this, we might limit the amount of golden rice that is being exported to those countries. And finally, socio-economical factors. How do we ensure we have equal distribution of the goods, of the products to everybody who needs it? Okay. Um, by maintaining a fair price, for example, and to make sure that, even people who cannot afford it will have access to it, right? So these are some of the challenges, right? My personal opinion, sure, these are important things to consider, but the advantage of the golden rice seems to outweigh some of these concerns. Okay. Anyways, what are we talking about? Right? How do we get end up at this topic? Well, just to remind you, we are talking about recombinant DNA, which are DNA that consists of genetic information from multiple sources. And the example of golden rice was an application of recombinant DNA in agriculture. Do we have any questions before I move on to the medical application? Excellent. Before I forget, I have a meeting on Wednesday, and I won't be able to make it until 1 o'clock. So we're going to be starting a little bit later on Wednesday, just this Wednesday only. Okay. Instead of 12:30, we are going to be starting at 1. If I'm a little bit late, don't go anywhere. Just stay there. I will show up definitely. But like I said, right, I'm hoping the meeting is not going to run over time. And then I will come here as soon as I can, right? But we're starting at 1 o'clock. I'll send you an email to remind you, but I'm sure you'll remember. Next, we're going to talk about the medical application of what did I say? Recombinant DNA, right? So insulin. Does anybody know what part of the body makes insulin? What organ makes insulin?
Pancreas.
Excellent, right? The pancreas makes um, insulin. And in case you don't know where the pancreas is, the pancreas sits right behind your stomach. Your stomach is around here, okay, on the left side, and right behind it, that is where you will find your pancreas. Okay, so it makes amongst other things, it makes this hormone called insulin. And in type 1 diabetes, which sometimes is called the juvenile diabetes, you'll learn about this next semester, people do not make insulin anymore, right? And without insulin, you cannot regulate your blood sugar, and your body basically thinks you're starving all the time, and it start eating away your proteins and lipids in your body, and it could be very devastating for the body. Okay, and so prior to the 20th century, the main commercial source of insulin were taken from the pancreas of cows and pigs. And much of the work were done by these scientists here, Frederick Banting and Charles Best. They are Canadian scientists. There's actually a building that was dedicated to them. That is dedicated to them in U of T downtown. Okay, it's called the Banting and Best building. People have classes in there. If you ever go downtown, you can visit it if you want. But they did a lot of study with insulin, and like I said, the primary commercial sources were taken from cows and pigs. So I want to open up the discussion a little bit here. What do you think some of the disadvantages are by using insulins from animals and giving it to people? Like what would the concern be, right? If they were to give us animals derived insulin? What would you be worried about, if anything? Okay, people might say harming the animals. Sure, yeah, that's a valid point. But what about from the perspective of people, from the person who are receiving it? Right? Are you going to be worried? No, no worries. Just give it to me. I need them. Okay, thank you. Okay, cross contaminations, right? Diseases, right? Like it's like you're purifying from these animals. I would be concerned about how pure is the extraction, right? If it has contamination, it could cause me to become more ill, right? There is quite a bit of cost as well associated with it. Like how much can you extract from one pancreas, right? Like if you need to produce it in large scale, right, it might not be possible uh, without substantial investment from that end. And also, if it's taken from pigs, then it might not be suitable for everybody, right? Because of religious beliefs, for example. Okay, so there are like a bunch of things that are not ideal. Okay, and then how do you know the animal version is effective in the humans? Okay, like it's not 100% identical, right? So there might be some efficacy issue there. However, that's all they had at the time, and it worked for a while until the late 20th century, where the creation of recombinant DNA allows mass production of insulin using bacteria. So basically, we take the insulin gene from human, we put it into the bacteria, and we turn the bacteria into insulin factories. Okay, so this is the commercial bacteria produced insulin. It goes by the trademark here, Humulin. Okay, it's as in like insulin for humans. That's what it is. And I am going to go through the steps that are required to create the insulin factories in bacteria. Okay, again, this is an example of a recombinant DNA application because we are combining human DNA with bacterial DNA. So we have four steps here. The first step is to locate the gene of interest. Right? So we understand all the proteins that we produce has to come from DNA somewhere. Okay, there is the instruction to produce the proteins, with the codons and everything, all that is within the DNA. Okay, so we have to figure out where the gene is located in the entire genome. And we have 23 pairs of chromosomes. Which chromosome is the gene on? And where is the exact location on the chromosome for the gene? We have to find it. Once we found the gene, we have to clone the gene. We have to make many copies of the gene. Then we have to insert that gene into a vector. A vector is something that will help deliver the DNA into the bacteria. And then afterwards, you just let nature do the rest. Right? So let's see if we can go through some of these things today. This is a agar play, agar, Agar, agar play. That's what you use to grow bacteria on. Each of these little circles, that's a colony of bacteria, right? And in this particular picture, the the bacterial colonies, some are white, some are blue. And this is like one way to determine whether or not your bacteria has accepted the vector. Okay, because I mean, look, you can't tell, right? Like if you just look at two bacteria under the microscope, even you can't tell which one has accepted the human DNA and which one hasn't. Okay, so there are techniques you can use to cause the bacteria to turn blue when they have accepted the human DNA, and if they haven't accepted it, then they remain white. So if we use that particular method, we can just scoop this out. This is like Jello, okay? So just like you can take a spoon and scoop up a little bit of Jello, you can take a like a wire with a loop at the end, and you literally scoop up this whole circle right there, which contains, thousands and thousands of bacteria with the human DNA in it. And then you can grow them in another plate and mass produce it. So you only grow the blue ones and not the white ones. Okay. So that's one technique of how to distinguish which bacteria has accepted the human DNA. Yeah. We're not going to be talking about this technique though, because that one is actually really hard to understand. We're going to be using another selection method. But, I thought I'd show you a picture like that. Okay, so we are going to learn how to make bacteria that can produce insulin. First step, locate the gene of interest. To locate a gene of interest would have been a very, very difficult thing to do 20, 30 years ago. Okay? Because back then, people have not fully mapped out the entire human genome. Okay? The Human Genome Project, HGP, Human Genome Project, um, started in October 1st, 1990. Okay, 1990. And it was an international collaborative project with the goal to completely sequence the entire human genome. Human genome is all the genetic information in the human, right? So starting at chromosome 1 all the way to chromosome 23, they want to map out all the ACTGs, right? And that took a while to complete. Okay, as you can see over here, it started at 1990, but it didn't finish until 2003. Okay. So when I was in high school, I actually presented the Human Genome Project as my research project for the course, for my biology course. Okay, and back then they just started. Okay, and now as you can see, it has been completed for more than 20 years, and all this information is available online. All this information is available online. Okay? So you can play around with it. This picture is a link. You can click on it, and it will take you to this uh, genome browser. Okay, genome browser. It's maintained by the University of California in Santa Cruz. And you can not only can you look up humans, you can look up other stuff as well. We have mouse, rats, zebrafish, and a bunch of other stuff. Okay, and here you can type in the name of a gene that you want to look up. Okay, so you can look up the insulin gene, for example, if you want. But can anybody give me a gene that we learn in the course? Okay. There's another one that's related to cancer. Anybody remember what it was called?
Oh,
That's right. Thank you. Okay, it's the BRCA1 gene. Okay, so if you key in BRCA1 gene here and then you click go, then it will tell you the BRCA1 gene is located on chromosome 17. Chromosome 17. Now, this is the centromere right here. And the shorter arm is called the P arm, and the longer arm is called the Q arm. And on the Q arm, there are like different sections, right, separated. So the BRCA1 gene is right here at this location. In fact, if you want to be precise, you can tell that the BRCA1 gene actually goes from, it tells you which base pair we're talking about. It is right here. 43,044,295 nucleotides to 43,125,364 nucleotides. So that's the exact location of the BRCA1 gene. Okay, and now we know where it is. You can actually click into the gene of interest. Let's see if this is going to work. Sometimes it's really slow while I'm doing the Zoom and it might not work. Let me just refresh the thing right here. Oh, there it is. Oops. One more time. Okay, right here. And we can see the genomic sequence. If I click into it, there are some parameters we can play with. Like, do you want to include the promoter? Do you want to include the introns? I'm not going to change any of them. I'm just going to click submit. And there you have it. This is the BRCA1 gene. Okay, is GCT, GAG, and then the whole gene. Look there. It's just pages and pages and pages of ACTs and Gs. This is the gene. This is the instruction to make the BRCA1 protein. Okay, and that's what our DNA is. We learned this, remember? You can actually change this into RNA, and then you can decode it. You can decode what CTT codes for, right? And it builds the amino acid for you. All right. And here you can kind of see there are some capital letters and then there are some lowercase letter. The capital letters are actually the introns that will be removed in the process of post-transcriptional modifications, and it's only the exons that will be left, right? So this is like real stuff that we've learned in the course. And you know, it's all on the internet. People can find whatever gene they want using the genome browser. Okay, so there it is. First step done, right? We've located the gene of interest. Then what are you going to do with it? Well, the next step is we have to clone the gene. We have the gene now. Okay, we know where it is. It is on chromosome 17, I think, and we have to make a lot of copies of this gene. We don't want to make the copy of all the DNAs. We only need this gene because this is the gene to put into the bacteria. Although I misspoke, we are not cloning the BRCA1 gene in this case. We are cloning the insulin gene. So we would have to find the insulin gene, I figure out where it is, and then we will clone the insulin gene. Okay. So how do we clone the insulin gene? To clone the insulin gene, we use a technique called PCR. PCR stands for polymerase chain reaction. Okay. Uh, Ordinary, regular, non-science people probably have not heard of PCR before the pandemic. However, during the pandemic, a lot of people needed to do a PCR test to see if they have COVID or not. Okay. So actually, PCR has been around for quite some time. Yeah, for like at least 30, 40 years prior to the pandemic. So it's not a new technique, but it's a technique that can be used to basically clone DNA, and you can do it in a very precise manner in the sense that you can decide exactly which part you want to copy, and you can do it within a very short period of time, and you can do it with a small amount of sample. Okay, so let me say it one more time. You don't need a lot of sample to start with. You can do it with one cell. Okay, if your technique is good, you can do it with a single cell. Okay, so like if they recover some blood from the crime scene, if they recover some semen from a crime scene, they could use that and use PCR to magnify the sample. They could do it from a hair. They could do it from like mummies or like fossilized organisms. You don't need a lot of cells to begin with. That's what I'm talking about. Okay. And you can copy the gene and make literally billions of copies within 2 hours. Okay? 2 hours. So that's what PCR is. Now I'm going to talk about how PCR works. Right? This is a PCR machine. Obviously, it's very expensive, not something that we would have lying around at home. But this is the reaction chamber. You're gonna have these tiny. A lot of the stuff I'm gonna be explaining to you are gonna be happening in these very tiny reaction tubes. Okay, these tubes right here. Okay, they are no bigger than this. Okay, kind of like this candy right here. They're smaller than this, and all the reactions is gonna be happening in them. They are called Eppendorf tubes. Okay, the names are not important, but remember, all the reactions that I'm going to be explaining happens in these small, tiny tubes, and you're gonna put them into the PCR machine, and you can program the machine differently depending on what you're trying to do, and basically it will double the amount of genetic material per cycle. So if you start with one copy, after one cycle, and I'll tell you what a cycle is, okay? But after one cycle, you will have two copies. And then after another cycle, you will have four copies, 8 copies, 16 copies, 32 copies. It doesn't sound like a lot at the beginning, but what if you do 30 cycles? That's 2 to the exponent 30, okay? I'm going to plug this into the calculator. If you have it, feel free to do it with me. 2 to the exponent 30, according to my calculator, is 107374184. Okay. So that's like a billion copies right there. Okay. And 30 cycles usually, like I said, take around 2 hours. Okay? Take around 2 hours. So how does it work? Well, PCR is closely related to the process of DNA replication. And that's what I was trying to tell you at the beginning, right? Like, what is biotechnology? Biotechnology involves manipulating biological processes, in this case, DNA replication, so that we can have it do what we want. Okay? So we are manipulating the DNA replication process so that we can clone, we can massively produce um, a section of DNA that we are interested in. Right? And the section of DNA that we are interested in this case, would be the gene for insulin. So, what do we do? Well, in cells, DNA strands are separated using gyrase and helicase. We learned this in lecture 6. But there are no gyrase and helicase in PCR. Instead, in PCR, DNA strands are separated using heat. And you basically heat up the reaction tube to around 95 degrees Celsius, and the heat will break the hydrogen bonds holding the two strands together. Depending on the CG's content and the AT contents, you might have to change this number a little bit. Okay, you might, if there's a lot of C's and G's, you might need to heat it up a little bit higher. If there are a lot of A's and T's, then maybe not as high. Okay, there's actually a formula that you can use to figure out what's the optimum temperature. Okay? Once the strands are separated, each strand can be used as a template to build the complementary strand. And we know that already. We understood that already. Okay. Now, how do you control where to copy the DNA from? Okay, so when you supply the sample, and if it comes in the form of a cell, then the full set of DNA is there. So how do you specify which part of the DNA you want to copy? Right? Like I said, we located the gene. Let's say the gene is on chromosome 14, right? At the tail end section of the chromosome. How do you specify that specific location? Well, what you need to do is to design primers. Okay, design primers. So what primers are is like this, okay? So let's say I have a textbook here, and then I want you to photocopy some pages for me, okay? But I worry you're gonna forget the pages. So what I can do is this. I can put a sticky tag. Okay, I'll say, put a sticky tag right here, and then let's say I'll put another sticky tag here. All right, let's waste another one. Why not? I'll put another sticky tag right here, and then I give you the book, right? So when you go and photocopy this for me, you will know, oh, okay, I have to start at this page with the first sticky tag, and then I'm gonna photocopy until I get to the second sticky tag right here. Okay, so that's basically what happens. Okay, what happens is the primers will help guide the DNA polymerase so that it knows where to start and where to end. Right? It's similar to what we learned in DNA replication. Right? The primer helps position the polymerase, right? Because polymerase, as you know, cannot initiate the process. It needs the primer to tell it, right? So rather than having a primase to create the primer, you create the primer. You design the primer. And so, I already have the sequence of the gene. Again, I know this is not the insulin gene, but you get the idea, right? And if we have a primer that would bind to the beginning here, okay, and then I would design a primer that would bind to the end here. Okay, I'm just doing the introns right here. That would bind to the ends right here. Okay. Then I know the DNA polymerase will know to only copy between these two sections. Everything in between is going to be copied. Okay, and I know how to design the primer. If I want to design a primer here, I would just use the complementary string. Okay, it would be like, G, 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 C, and then A, G, so on and so forth, right? In fact, nowadays, companies do this for you. You just tell them which gene you want to copy, and then they will even design the primer for you. You pay one fee, and then, they would even clone it for you. Okay, like people don't sometimes they're not going to clone it themselves, right? They just outsource it because it's cheaper and you save some time that way. Anyways, um, so to actually copy the DNA, you would need to have the DNA polymerase, right? But we actually cannot use DNA polymerase 3 here. Okay. DNA polymerase 3 is like the human ones, right? The one in yeast as well, right? But we cannot use DNA polymerase 3 in PCR uh, because we are heating the sample up to 95 degrees Celsius. To separate the DNA, I need to heat it up to 95 degrees, right? At that temperature, you're going to denature the protein. Okay? Remember, DNA polymerase is just a protein, and if it's too hot, it's going to unfold, it's going to be denatured, and it's going to be non-functional, right? So what do we do? Well, it turns out scientists have discovered bacteria living in hot spring. Right? This is a picture from like Yellowstone, and they have a lot of these like hot spring geysers. Okay, and the temperature goes as much as 110 degrees, 120 degrees, really really hot. But certain types of bacteria can live in there. Right? And what scientists did was they took the bacteria and they studied them and found out they have a special kind of polymerase that is heat resistant. Okay? It's called tag polymerase. Okay? So this is the polymerase used by those bacteria to copy their DNA, and they are fully functional at really high temperature. So in PCR, we are using tag polymerase instead of regular DNA polymerase 3. Okay, did you follow the story so far? Right. Let's quickly recap. We identified a gene of interest. We designed primers to know where to copy it. Okay, and then we're gonna separate the two strands using heat so that I have the template. And then the tag polymerase is gonna be making the copies of DNA for us. Questions before I move on? Excellent. So here we go. This is the gene of interest. We're gonna separate it using heat. The primers will bind to the location of interest, just like the sticky tags I showed you, and then the tag polymerase will copy. It will copy. Okay, so now after one, this is called one round, I will have two copies. I started with one copy. After one round, I have two copies. Okay, so typically there are going to be um, three steps involved in each cycle. I'm just going to copy this and put it right here. And then I will post the notes for you afterwards. Easier that way. So here, denaturation. What is denaturation? Denaturation is to separate the double helix using heat. Okay, so like I said, around 94 to 96 degrees, depending on the GC content of the DNA. And then you want to cool the temperature a little bit. You want to lower the temperature a little bit, not all the way back to room temperature. If you do, then everything is just going to come back together. You want to lower it a little bit to around 68. Again, these is just like, don't memorize the numbers. The temperature doesn't, like it's, like it depends. Different sample requires different temperature. These are just rough estimates, okay? But by lowering the temperature a little bit, this annealing stage allows primers to attach. Okay, you need the primers to attach so that you can specify which section of DNA you want to copy. Allows the primers to attach. But this temperature is too low to activate the tag polymerase. Remember, tag polymerase are taken from hot spring bacteria that lives in environment that is at hundreds of degrees, right? So 68 is actually too low for it, and the enzyme does not work. It's not going to be denatured, but it's not going to be working. So you will need to heat it up a little bit, okay, to roughly 72 again. So this activates tag polymerase and trigger the copying. You don't want to heat it all the way back up to 94 because then everything would separate, including the primers. It would be like ripping out the sticky tags after you put them on. You don't want that. Okay, so you just heat up a little bit. So over here, let's take a look at the picture. We have the original DNA sample. We have the primers shown here in pink, and then these are just the nucleotides. Okay, so step one, heat will separate the strand. Step two, annealing. You allow the primers to bind. And step three, we're gonna clone it. Okay. And then now we have two copies. And you repeat, and you have four copies. If you repeat again, you have eight copies, and so on and so forth, and so on and so forth. All right. We good? So we located the gene of interest. We cloned the DNA. We made, millions of copies of it. Okay. With 30 cycles, you have billions, right? As long as you have the nucleotides around, you'll be able to keep on doing this. Okay. Um, and then what's the next thing? Insert the DNA into a vector. But before that, let's do a poll question. Let's see what you think. Go ahead. All right. Thank you for those of you who participated. Let's take a look here. You are doing 4 cycles. So that's 2 to the exponent 4. Each time you do a cycle, you double, right? So that's going to be 1. So that's going to be, sorry, 16. Okay. 2 times 2 is 4. 4 times 2 is 8. 8 times 2 is 16. So that's if you start with 1 copy, but we started with 10 copies. So 10 copies times 16, 160. Okay. So basically, it's like the starting number times 2 to the exponent of n, okay? Where n is the number of cycles. Number of cycles. And question. Okay. Um, I was gonna go through the COVID-19 tests with you, but maybe we'll skip that. Okay, maybe we'll skip that for next time. It's not gonna be on the test, but we're a little bit tight on time, so we'll skip this here. Okay, but it's a different kind of PCR that they do. Okay, it's called RT-qPCR, but maybe another time. All right, next step. Okay, so we identified a gene. We know where it is. We cloned it. Now we have millions of copies of the gene of the insulin gene sitting in the vial. Now we want to put this into a vector. The vector will be fed to the bacteria, and then we'll move on from there. Okay. So to understand that, we're gonna take a closer look at bacterial DNA. So bacteria, they have their core DNA. Okay, this is core bacterial DNA, which is of course not in the nucleus. They just like kind of uh, located in like a central area. So this is the core genetic material. And in addition to the core genetic material, there are these extra circular DNA called plasmids. And the plasmids usually give them some kind of special properties, if you would. Okay. So some plasmid allows them to create a thicker shell outside of the cell wall so that they're more resistant to dry environment. Okay. Sometimes the plasma would give them coatings that allows them to stick to each other better. So it forms a more resilient biofilm, is called. Sometimes the plasmid is going to give them the ability to destroy antibiotics. Okay. So the point is, plasmids usually give them some survival advantage. And what happens is two bacteria can come close to each other and then they could swap plasmid or they could share plasmid, right? So like I don't remember if I told you this before. It's deja vu. Sometimes you teach the same thing too many times, then you forget which class you told what. So forgive me if I'm repeating myself. So let's say there is a bacterium here and it has a red plasmid, okay? The red plasmid allows it destroy to destroy red antibiotics. Whatever the red antibiotics is, like amoxicillin or something. Okay. So if you get infected by this bacteria, and you take this antibiotic, it's not going to do anything for you. Okay. You're going to continue to be sick because the bacteria is not affected by it. They have this special plasmid that makes it resistant to the red antibiotics. Okay. Antibiotics. And over here we have another bacteria which happens to have the blue plasmid that makes it resistant to the blue antibiotics. Antibiotics. Okay, so you cannot kill it. Okay? So I want to ask you a question just to make sure that you are with me. If I'm infected with the red bacteria, okay, I'm coughing like crazy, okay, and I need antibiotics, should I take the red pill or the blue pill to get better?
Video one?
Blue.
The blue pill, right? That's
Yeah.
right. I need to take the blue one because the blue one, this bacteria is not resistant to, right? Okay. Similarly, if I get the blue bacteria here and I'm infected with it and I'm so sick and I need antibiotics, I'm gonna take the red one. Make sense? Okay. Here is where things get a little bit scary. Okay. So let's say you got infected with the red bacteria, okay, and I got infected with the blue bacteria. We are both very sick, okay? We need to see the doctor. So you went to the walk-in clinic and I happened to go to the same walk-in clinic, okay? And then, let's say while I was waiting, I decided to, look at some magazines or something. Okay, so I pick up a magazine and then I cough into it. Okay, so now I cough some blue bacteria onto the magazine. Okay, so there's the magazine. Okay, this is a really real possibility. Okay, like don't think I'm just making it up. Okay? So now I got some blue bacteria there. Okay, and then I put the magazine back because it's my turn to see the doctor. Anyway, so I get my antibiotics. I went home, and then you were waiting for your turn. You pick up this magazine, okay? And let's say you are also coughing, okay? So you cough, 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 and then you get some red bacteria onto the magazine. It doesn't have to be the magazine, right? You could be coughing into your hand and then go touch a doorknob, right? And this would be happening on the doorknob, right? Okay? And then this is what's gonna happen. The two bacteria now they found each other. They're like, hey, let's exchange the plasmid. Why not, right? So then they're gonna do it's called bacterial sex, okay? Conjugation, that's what it's called. So they have a little thing that they can extend and they will connect each other. It's called a pilus. And what happens is the blue bacteria will clone the plasmid and then it will send a copy over. It's like, hey, I got this blue plasmid. I'll give you a copy because it's so cool. And then the red one is like, you know what? I'm gonna give you the red one in exchange. And then they separate afterwards. Then all these bacteria which engage in this particular activity will now become a new kind of bacteria where previously only resistant to a single antibiotic. And now what happens? What do you think happens? Right now, they will both be I can't copy it, but they will both be resistant to both kinds of antibiotics. Okay? So you and I went home with our drugs. Okay, we'll get better. But like some other person who was there for some other reason, if they decided to pick up the magazine, they decided to touch the doorknob, and then they didn't wash their hands, and then they touched their face after, they could pick up these newly formed, newly created bacteria with the two of the plasmid. And then they would get sick. They would get sick, and the red antibiotic would not help, the blue antibiotic would not help, right? And then they would need like some other kinds of antibiotic. And this process happens in real life, okay? And not just for two antibiotics, but for like many different kinds of antibiotics. So you can imagine meeting a third bacteria, and this third one is resistant to like a green antibiotics, and then they go exchange it. And over time, you're gonna create a bacterium with so many different kinds of plasmid that makes them resistant to a whole bunch of antibiotics. We call them superbugs. Superbugs. You might have heard of that on the news before. Okay. So superbugs are resistant to a wide group of antibiotics. Okay. Like it usually are resistant to all of the versions of penicillin. Like, there are different kinds of penicillins with different strength. Okay. And if you get sick with one of these things, it's really, really hard for you to recover because like you literally have no drugs to help you. Okay. So there are two very concerning kind of superbugs at healthcare setting. One of them is called MRSA, the other is called VRE. Okay. MRSA is methicillin resistant S. aureus. You can just look these up if you're interested, okay? S. aureus lives on you, on the surface of the skin, but they are not antibiotic resistant. They don't usually cause problems. But if they meet a resistance one at the hospital, okay, they could exchange plasmid and mix it into a serious infection, Okay. Same thing with VRE, which is vancomycin resistant enterococci. Both of these are very scary at the hospital setting. So if you are admitted to the hospital, they often swab you to make sure that you don't have these. Okay. They would put you in a isolated room, swab you, make sure that you don't have them before they move you to like a share room or something. Okay. If you have these things, you will be kept in isolation because they don't want it to spread in the hospital. All right. Um, so uh, basically, my point is the plasmids give bacterial advantage, and that's why bacteria likes to pick up plasmids whenever they have a chance. Okay, they don't have to get the plasmid from another bacteria. Okay, if a bacteria dies and leaves behind some plasmid on the surface, and another one comes along, they could just scoop it up. Okay, so there is a tendency for bacteria to just pick up plasmid from the environment. So understanding that allowed scientists to create a recombinant DNA. Like, what if we take the insulin gene, we put it in a plasmid, and then we just leave it in the environment and then expose the bacteria to them? Then the bacteria is going to pick up the plasmid. The bacteria doesn't know what kind of plasmid is it. It doesn't know if it's going to give it antibiotic resistance or if it's gonna make it produce insulin, right? It doesn't care, it doesn't think, right? As far as we know, they don't think, okay? And so they pick up this plasma with the insulin gene in it, then now they start to pump out a lot of insulin. Okay, so that's where the story is heading. But first, to understand how to put the gene into the plasmid, we have to understand something called restriction enzymes. Restriction enzymes. So let's take a look at this. Bacterial enzymes that cuts DNA sequences at a specific recognition site. Okay. Now, even though the bacteria likes to pick up plasmid from the environment to enhance their adaptability, their survivals, for example, they don't actually like to have foreign DNA being inserted into them. Okay. And a while back, I was showing you the bacteriophage, with the experiments stuff, right? Hershey and Chase experiments. So when a virus injects DNA into a bacteria, the bacteria has a defense mechanism to cut those DNA up, to shred them. Okay. And these enzymes are called restriction enzymes. Okay, so normally what happens is this is bacteria. Let's say we have a virus that's coming in, and the virus is injected the DNA, and the bacteria doesn't like that. So it's going to take the restriction enzyme and it's going to shred them up before they could, hijack the bacterial machinery. Okay. So these restriction enzymes, they are basically like scissors, okay, for the bacteria to chop up foreign DNA. They're not going to chop up plasmids for reasons we're not going to go into. They're only going to chop up the like the viral DNA in this particular case. And how they work is they typically recognize a special sequence that's about 4 to 8 base pair long. And this sequence usually has a property that makes it what's called complementary palindromic sequence. Complementary palindromic sequence. So, what is a palindrome? A palindrome in English is a word that spells the same forward and backwards, or it could be like a short sentence that's the same forward and backwards. So, like madam is a palindrome. Okay, M-A-D-A-M. If you read it backwards, it's also M-A-D-A-M. Right? Level is a palindrome. Wow, kayak. All these are palindromes. Okay, and this is a really long one. Was it a car or a cat I saw? If you read it backwards, was it IT a cat or a car I saw, right? It's like the same thing, right? So that's what a palindrome is, same forward as it is backwards. So the restriction enzymes, the defensive scissors that the bacteria have, they don't randomly cut foreign DNA at random locations. They cut them at these palindromic sites. Okay, these complementary palindromic sites. For example, we have a restriction enzyme here. It is called eCore R1. They have really weird names, okay? The restriction enzymes are named after the bacteria that you take them from. So eCore R1 is taken from E. coli. Okay? Not too important. But what's important is if you have a sequence here, where is it going to cut? Well, it's going to cut at this location here. It's going to make a zigzag cut because this orange section, excuse me, is a palindrome. It's a complementary palindrome. If you read it on one strand, GAATTC, it is the same backwards on the opposite strand, GAATTC. Okay. It's a little bit different than English palindrome. I'm not saying it's the same forward and backward on the same strand. I'm saying it is the same forward and backwards on the complementary strand. You understand the difference? For example, like this, TTA, TT. This is not a palindrome for the restriction enzymes, even though it reads the same forward and backwards, but that's not what I'm talking about. I'm talking about forward the same and backwards the same on the opposite strand. So what would the opposite strand be? If something is TTATT, the opposite strand would be, someone help me out here. What's the opposite strand?
A A T
Yeah.
A A.
That's right. So you see, if you read it forward, it's not actually the same as backwards, right? So that's not a palindrome in the context of restriction enzymes, okay? It's only a palindrome if it's the same forward and backwards on the complementary strand. Okay? And then it's gonna cut it. It's gonna cut it like that, right? So each restriction enzyme will have its own recognition site. Okay? Equal R1 is always going to recognize GAATTC and is going to cut it zigzag like this. Okay, there's another one called small one, which is taken from this bacteria. You don't have to memorize the names, okay? And it will recognize the GGGCCC and then it will cut it straight down. Okay, if you have GGGCCC and then you expose it to equal R1, nothing will happen. Okay, these scissors will only cut at their own recognition site. Okay. It's not going to be effective for other recognition sites. Okay. You don't have to recognize, you don't have to memorize the recognition site. I will always give you the recognition site in the question when I need you to use them. Okay. It's not even going to be like a separate tab. It's going to be like right within the question. Now, the restriction enzymes can cut in one of two ways. They could cut a zigzag like this, or they could cut straight down. Okay, when you cut it straight down, like this, and we call these blunt ends. And then over here for the other ones, like this one, and this one, where is zigzag? We call these sticky ends. Okay, and for the purpose of putting a gene into a plasmid, we want sticky ends. Okay, blunt ends are not useful in our discussion. They're useful in some other contexts, but not what we're trying to do. And what we're trying to do is put the human insulin gene into a plasmid and then feed it to a bacteria. So why are they called sticky ends? Because after you do the zigzag cut, this AATT portion that's hanging is actually complementary to the TTAA. So there is a tendency for them to stick back together. Right? It's like Velcro. After you peel them apart, they could just easily stick back together, but not the blunt ends. Okay, blunt ends, there's nothing hanging out. Right? You can't really glue them back together. Hey, questions?
In number three, is there any mistake?
Mm, which one?
Uh,
Here? Here?
T C G A below number. Oh no, four.
This one?
Yeah.
What's the mis? Oh, yeah. Wow. No one, all this time, nobody has ever picked this up. Very good. Okay. This should
C.
be a C. Yeah, there you go.
Mm.
Very good. Wow. Good eye, good eye. Okay, so there you go. That's a mistake right there. I haven't noticed all these years. And no one has either. Very well. Okay, are we together with the restriction enzymes? Yeah, I know I'm taking your break away, but I'm almost done. I just need to go through the slides with the restriction enzymes, and then we're gonna call it. All right, so here is small one, SMA1, small one. Uh, this is a Roman numeral one as opposed to I. Okay? And the recognition site is right here, GGGCCC. And it's going to cut straight down like that. It's going to create a blunt end. So pay attention to the 5' and the 3' over here. And I want you to look at the sequence and I want you to figure out where are the cuts going to happen. And when you figure out how many times it is going to get cut. Okay, so I have some answers coming in. People saying it's going to get cut twice. Anyone else? Okay, so let's take a look. Over here, we have 3' here, so I'm looking at 3' and 3'. 3' is CCCGGG. So basically, I'm gonna look for CCCGGG right there. That is the CCCGGG, and that's gonna be our first cut right there. I will keep on looking. Keep on looking. There it is, another CCCGGG, and we're gonna make another cut right here. So two cuts. Two cuts. How many fragments with two cuts? One, two, three. Three fragments. Okay. You take a piece of paper, you cut it twice, you get three pieces, three fragments. Is everybody okay? Do you need me to explain this? Like, let me know if you get confused, because it's only gonna get more confusing as we progress. Are we okay?
If we get here GGGCCC, if it is only 2G, 2C,
Yeah,
then
then
it.
it doesn't count. Yeah, it has to
Mm,
be it has to match exactly.
okay.
Very good. Okay, identify a palindromic sequence. Okay, so the palindromic sequence should be like four to eight, um, 4 to 8 nucleotides long. Okay, the easiest way to do it is probably just like um, list it all out, like write down the complementary strand here. I'll do it right on the thing right here. Okay, so I'm gonna let me see if I can put this at the background. Okay, and I'm just gonna type it out. Now you can do it as well. Oh, looks like I can't type it out. I'll just have to do it a little bit lower here. Um, okay, so like we're gonna basically um use the base pairing rule. Please check it over when you're doing it. Okay, so it should be something like this. I have a feeling it's a little bit bigger, perhaps 12. There we go. Okay, I'm just gonna put it right there below it. Okay, maybe that's too big. Let's try 11. Now it's too small. Doesn't matter, I guess. Let's try 11.5. All right, there you go. Just right. All right. So we're gonna see if we can identify any palindromic sequences. Do you see any? Well, I see one right here. See one right here. This is TTTAAA, right? Right there. TTTAAA. So it's the same forward as it is backwards. That's one palindrome right there. And do you see another one? Okay, might be a little bit harder to see because it's looks like it has been shifted a little bit. But anything else that you notice? Okay, right here, that's another one. Again, it's a little bit slanted. I apologize for that. But if you read it, it's the same. Sorry, right there. So this way, GAATTC, and backwards is also GAATTC, right? So you might be able to find a few more if you look closely. But basically that's what you have to do.
Is there any technique to find out quickly?
No, no. But like I'm not gonna be like this on the test, okay? Because it will be very time consuming, I understand. Okay? So on the test, I will probably just give you short sequences like A, B, C, D, and then I'll say which one is palindrome. So that way you can figure it out like relatively quickly, right? Like rather than giving you a whole strand and making you write it out. But the point is, do you guys understand like like what it is? Would you recognize it if you see one, right? I guess that's my point. And I hope the answer is yes. Can you guys create a palindrome maybe? Yeah. Give me a palindrome that's 6 nucleotides long. 6 nucleotides long. Right? So create your own. I'll give you a minute. And just type it in the chat box when you have it. And we're gonna move on from there. We're almost done. Okay, just one more slide, guys. Okay, so we have one from Samuel, which is GCGCGC. Yeah, that's correct. So he's playing it safe. He's just using two different letters. Okay, and this is definitely a palindrome. Okay, anyone else before I tell you the secret? Once you know the secret, this is so easy. There's like anybody can do it. But does anybody have another example that they want to share with the class? Okay, that's fine. I'll tell you the secret. If we do 6 nucleotides long, then you start with 3 nucleotides. Any three you like, okay, whatever you want. Okay? So, I'll do TTC. Okay, TTC. TTC, like the like the rockets, okay, TTC. Then what you do is on the other side, you also put TTC backwards. Okay, so at least TTC here and then TTC backwards are the same. And then you just fill out the rest using complementary. So T goes with A, T goes with A, and C goes with G. And then here G, A, A. You see? That's it. Easy as that. Do you understand? Okay, so make another one. Okay, show me that you understand. Make another one and share with me. You don't have to type both strands, okay? Just type me one strand and I'll know if you're right or not. Okay. Hopefully I get at least three answers from the class, and then we'll do one more slide and we're gonna go home. All right, we have another one coming in, which is TTA. Okay, so after you start with TTA, don't forget you should move to the other side and do TTA as well. All right. Then you just fill in the rest. AAT, TAA. Okay. Anyone, anyone else? Like maybe one more? Or you guys have had enough? If you don't understand, you should ask, right? It's not gonna go away. Hmm. Athira has another one. Um, GTC, GAC. Yep, that is exactly it. Okay, 100%. There you go. All right, that's it. Okay, we have one more question, and then we're gonna stop. There we go. This is another DNA sequence, and we are treating it with two restriction enzymes with equal R1 and HIND3. Okay, HIND3, that's how you pronounce it. HIND and then D and then 3. So how many times will we cut it and how many fragments will be produced? Okay, I have a few coming in with the palindromic. Yep, they're all correct. Okay, yeah, good, good, good. So over here, look for the restriction site, look for the recognition site for the restriction enzyme equal R1, and then look for another one for HIND3. All right, so for equal R1, I'll use red for equal R1. It is GAA-TTC 5 prime to 3 prime. So 5 prime to 3 prime. I'm looking for GAA-TTC. So there it is right at the beginning, GAA-TTC. It's going to cut it right there. That's one cut. Do we have another GAA-TTC? Well, all you have to do is keep on looking. Keep on looking until you find another one. Oh, there it is, GAA-TTC. So that's going to be another cut. Okay, so two cuts from equal R1. I think that's it. There is no more from equal R1. But then we have Hindi3. Like if you're trying to pronounce these things, okay, just look for the italics So HIN is italics. So you say that hin, and then these are not italics. Then you say D3. That's what it is. So where are we going to cut with a hin D3? It is AAGCTT. AAGCTT. So AAG right there. AAGCTT. And we're going to cut it right there. That's it. That's it. So three cuts in total. Three cuts, which equals to four pieces, four fragments. Right? That's it for this lecture. Uh, I kind of know the answer already, but I have to ask. Okay, it's my responsibility to ask. Does anybody have any question from the study guide of lecture 8 with the genetics problem? You know, there's a lot of genetic problems in the study guide, right? Dihybrid, sex-linked. Does anyone have any questions from any of those? I can take them up now. I mean, I have like 20 minutes. Let me know if you have it. I'll give you a few minutes. Okay. If you gotta go, then you can go. Okay. Remember, class for Wednesday is going to start at 1 o'clock. Okay. 1 o'clock. Right. Okay. I'm gonna stop the recording. Stick around if you have questions for the genetic stuff. If not, I'll see you Wednesday.